And the next topic is understanding the differences between primary and secondary active transport. First of all, what is primary active transport? It's the movement of substances against their concentration gradient by directly using energy from ATP, from low concentration to high concentration. The classic example is the sodium-potassium pump, okay, that transports 3 sodium ions out of the cells, okay, and takes 2 potassium ions into the cells against their concentration gradients. Okay, we are going to see or study the example in more detail when we study the action potentials. Okay, it's very important to maintain these differences between the intracellular and extracellular compartments. Okay, the cells have many channels that are typically open. We call them leaky channels. So continuously potassium is escaping out and sodium is coming into the cells, but using those open channels. Sodium-potassium pump has to be constantly working, taking the potassium back into the cell and pushing all the sodium out of the cells. Okay, it's necessary for maintaining the resting membrane potentials of the cells, for example, in the neurons, any excitable cell in the body. Now, secondary active transport, okay, also moves, okay, substances against their concentration gradient, okay, but not directly using ATP, but using the concentration gradient that was created by the sodium-potassium pump or any other primary active transport mechanism. Okay, you can take, for example, glucose or amino acids, okay, and transport it against its concentration gradient. Typically, this transport has to be coupled to the transport of sodium, okay? But sodium is going to move, okay, following its concentration gradient that was created by the sodium-potassium pump. Remember that always the sodium-potassium pump is maintaining a very low sodium concentration inside the cell. Yep. While there is this low sodium inside the cells, sodium may enter, and if we attach to the same transporter of sodium an amino acid or glucose, we are going to move the glucose and the amino acid or anything that we bind there, okay, against its concentration gradient. Okay, we're going to see that in a picture that is better understood when we see the pictures. And there are different types, okay. There are 2 big types. One is called co-transport, and the other is called counter-transport. There are different names, symport or antiport, we can use as well. Co-transporters or symporters move substances in the same direction, typically 2, sometimes 3 ions at the same time. In the case of the glucose-sodium co-transport or symport, we are going to move glucose into the cell against its concentration gradient. Imagine that the cell has a lot of glucose, and you want to put more there against its concentration gradient, and that is going to be coupled to the movement of sodium down its concentration gradient. Example of counter-transport, we have the sodium-calcium exchanger. Okay, we are going to move sodium following its concentration gradient, okay, but we are going to move calcium out, okay, against its concentration gradient. That sodium-calcium exchanger is extremely important, for example, in the heart, okay, or in any muscle cell. Okay, remember, the muscle contraction depends on the presence of calcium in the cytoplasm. If there is calcium in the cytoplasm, all of these mechanisms of actin and myosin are going to be activated. So for muscle contraction, it's great to have calcium, but when you are going to relax the muscle, you don't want to have calcium there. So for example, in every heartbeat, systole, diastole, you need calcium, no calcium, calcium, no calcium. So you need to make the calcium enter and disappear almost instantly. Imagine when you are exercising, 150 beats per minute, 160, how fast the calcium has to move in and out. So you need to, in the heart, okay, rapidly take the calcium that is inside the cell and take it out to the extracellular compartment or to the sarcoplasmic reticulum, but make it disappear so the heart can relax. And that needs ATP. So what happens if there is no ATP because there was no oxygen? Contraction, no relaxation. Okay, that's one of the things that where you can see the clinical application of this. So here we have a picture, a diagram, that shows one example. Okay, we have here, this is happening in the kidney. Okay, on this side here we have the tubular lumen, that is where the urine is. On the other side we have the interstitial space. On this corner here, upper right corner, we have the sodium-potassium pump taking potassium into the cell and sodium out of the cells, creating a very low sodium concentration inside the cell. That low sodium concentration, okay, will favor the entry of sodium from the urine into the cells, following its concentration gradient, and that sodium entry, okay, is linked, okay, to the entry of glucose into the cells using the sodium glucose linked transporter, SGLT2. And this is a classic example of a secondary active transport that is transporting two substances in the same direction. So it's called a symporter. Okay, and notice that sodium is going to be taken out by the sodium-potassium pump, and glucose then is going to go to the extracellular compartment using another glucose transporter that is called GLUT2. Once you have a lot of glucose here, glucose will go to the interstitial space and from there to the blood. And maybe some of you know that there are some medications that we use for treating people with diabetes mellitus, which function is to inhibit the sodium glucose link transporters. They are called SGLT2 inhibitors. Because remember, that is the urine. If we inhibit that transporter, glucose is going to continue. It's going to end up in the bladder and in the toilet, and it's not going to enter the circulation, so we help maintain the sugar levels low. Okay, that is one example of how all this may apply in pharmacology in the future. Because another way would be blocking the sodium-potassium pump, but I don't think that's too smart, no? Now there you have examples of, on the left side you have some co-transporters or symporters. Okay, there is one that is called the sodium-potassium chloride co-transporter. There are medications to block that. That co-transporter is in the kidney as well. If we block that transporter, what is going to end up in the urine? Sodium, potassium, and chloride. So we eliminate sodium chloride, we eliminate water. That contributes to lowering the blood pressure and to increase the rate of urine formation, and that is called a diuretic. Now, what is the problem? That we are losing potassium too. Sometimes people may develop hypokalemia or low potassium levels. When you study, please notice, I am not studying to remember this for a quiz or for a test, so I can forget about this and move on. You are going to be studying many medications. Inhibitors of the sodium glucose cotransporter, inhibitors of the sodium potassium chloride symporter, inhibitors of all these things that you see there. Okay? You can inhibit them, you can activate them so they work more depending on what you want to get. Okay? At the end, when you start practicing and you practice for several years, you won't even need to think about these things. Okay? Medicine will be like cooking. Let me put this to get that. Oh, it's too much. Let me put something to try to block what I did before. Okay, on the right side you have some exchangers or antiporters. Sodium calcium that we already mentioned, sodium hydrogen exchanger. Okay, notice that here you need to only remember the concept. In the notes you have an explanation of all these things, how they apply in pharmacology, how they apply in clinical practice. You need to study that now. Okay, just understand that you will be using this a lot in the future. And for example, this, as I told you before, explains the mechanism of action of the medication. You are eliminating sodium. Sodium eliminates water. Less sodium, less water means less. And the secondary effects of the medications. Okay? Now, regarding the bulk transport, we already mentioned the concepts. There you have a more organized description, okay, of these mechanisms of exocytosis, endocytosis. I'm more specifically talking about phagocytosis. Okay, but there is nothing new, okay, different from what you had in the previous table.